The presence of the Romaine hydropower complex changes the river's flow regime. Hydro-Québec will be conducting an environmental follow-up until 2040 to see how the milieu evolves and to assess the effectiveness of mitigation and compensation measures put in place. The follow-up includes a study of the softshell clam, a mollusk present in the mouth of the Rivière Romaine.
Softshell clams are found along the shoreline of the estuary and the Gulf of the St. Lawrence in areas subject to tides. It's an endobenthic organism, which means it lives buried in the substrate or in the seabed. Since softshell clams live in the substrate, they're affected by several parameters, especially when they're exposed at low tide. They might be exposed to wind, storms, ice. All these elements can change the substrate and therefore affect the softshell clams as well. We monitor softshell clams in order to characterize their habitat and the community as a whole, its density and yield, the age structure of the population, and other such parameters. Softshell clams are edible, so they're very important for local residents. Our follow-up area consists of 3 sectors. The first main sector is the mouth of the Rivière Romaine. To determine whether the project is affecting the softshell clam population in this sector, we're also using 2 control areas, the mouth of the Rivière Mingan and the Baie des Trilobites. We'll compare our data with results from these control areas and with the results of follow-ups from previous years, as well as a baseline study in 2013 that was carried out before the commissioning of the Romaine 2 generating station. To characterize the softshell clam population, we do tests on-site and in the lab. During the 2017 follow-up, we took 118 samples on-site. In other words, we gathered clams at 118 locations. We take a spade, we dig to a depth of 30 centimeters in a circular quadrant. We collect all the material and pass it through a sieve. All of the mollusks in the sieve, regardless of the species, are kept for identification and analysis. In the lab, we take several measurements: length, thickness, mass, and the length of the hinge ligament. We also count the growth lines on the clam shell to calculate the age of each specimen. Once all the analyses are completed, we return the live clams to the sampling site. We even bury the larger clams, which are big enough to be legally harvested, in order to ensure their longevity. Our results from 2017 show a slightly lower density of softshell clams at the mouth of the Rivière Romaine and also at the mouth of the Rivière Mingan. We didn't observe notable trends in the Baie des Trilobites because we didn't collect many clams in that area. Since the slightly lower density of softshell clams was observed in the natural environment at the mouth of the Rivière Mingan and at the mouth of the Romaine, we assume for the time being that this is due to natural variations. Future follow-ups will add to these findings and validate the predictions of our impact assessment.